Hi everyone, I hope I'm audible to you. So we have been doing molecular basis of inheritance. Today we will be understanding RNA and its types. Okay, so let's start with ribonucleic acid. So how is ribonucleic acid different from nucleic acids, right? So deoxyribose nucleic acids. So if you look at the structure of the pentose sugar, then in the pentose sugar there is an OH in this position which is not present. The removal of this oxygen, right, leads to the formation of deoxyribose, which is the pentose sugar which is used in DNAs. Also, when it comes to the nitrogenous bases, I think all of you know that uracil is used in place of thymine. Thymine is used in DNA, whereas uracil is used in RNA. Correct? So now if I were to classify RNA, then we could classify into 6 broad categories starting from ribosomal RNA, messenger RNA, transfer RNA, and then you have genetic RNA, small nuclear RNA and small cytoplasmic RNA. So let's look at these 3 first because these 3 are the least bioavailable. Starting with genetic RNA, these means RNA which forms so in organisms which lack DNA, and RNA is the genomic genomic structure. Okay, so RNA forms the genome. It's only found in viruses, almost all plant viruses, and very few animal viruses, which we call riboviruses. However, you will have to remember that RNA, when it forms a genetic material, it is not capable of translation. This RNA, when inside a host, has to get reverse transcribed into a DNA, and then from DNA, this whole of central dogma can happen. So from DNA, then it will get converted into mRNA, and from mRNA, it will get converted into a protein. Okay. So yes, they are the genetic material, but for them to be able to express it, they need to convert into DNA and then into mRNA and protein. So this process of converting RNA into DNA is known as reverse transcription. Why? Because DNA to RNA is transcription, so the ulta of it is reverse transcription. Now moving on, you have small nuclear RNA. Small nuclear RNA is found in the nucleus and it's called small because it is lesser than 300 nucleotides in the system. Okay? And it is very, very useful in functions like RNA and rRNA and mRNA processing as well as gene splicing. So what is gene splicing? So when we make our mRNA, it is not a mature mRNA, right? It's not an expressive mRNA because eukaryotic mRNAs have introns and exons. Introns do not express to give you a protein. So once the mRNA is made, the introns need to be cut out of it so that the exons can be joined. And then the joint chain of exons will be your translationally active structure. Correct? So that is known as gene splicing, and snRNA or small nuclear RNA helps in that as well. Okay? Coming to small cytoplasmic RNA, as the name suggests, they are very small and they occur in the cytoplasm. They are usually free occurring in the cytoplasm. There are multiple functions, but for NEET specific, you will need to understand one function, which is this. It is a single recognition or site recognition protein, which is produced by a very one group of small cytoplasmic RNA. What it does is, it helps in the ribosome endoplasmic reticulum binding, okay, and therefore helps in transcription. Yeah. Now moving on to the 3 most important types of RNA. Starting with ribosomal RNA. Ribosomal RNA forms the ribosome. So along with the proteins, the ribosomal RNA is embedded into the protein. rRNA is measured in Svedberg's units. Okay, so it's a sedimentation coefficient called the Svedberg unit, and therefore we denote it by S. If you look at the eukaryotic rRNA, there are a total of 4 types: 28S, 5.8S, 5S, 18S. 5S is common for both prokaryotes and eukaryotes. Okay. 5S is also unique because it is the only rRNA which is extranuclear in origin. All other types of RNA are nucleolar in origin, which means that it is produced in the nucleolus by the nucleolar organizer region on the norm. Except 5S. 5S is extranucleolar. Now, as you know, the ribosomes consist of a larger subunit and a smaller subunit. So, in eukaryotes, there are 3 subunits or 3 smaller fragments, 28S, 5.8S and 5S, which constitute the larger subunit, and 18S constitutes the smaller subunit. In prokaryotes, however, there are 2, 23 and 5, which constitute the larger subunit, and 16S, which constitutes the smaller subunit. Okay? Moving on to transfer RNA. Extremely important. So, transfer RNAs help in transferring their adapter molecules. They help in transferring the amino acid based on the codon sequence. Correct? So, if you look at the structure, right, the secondary structure is a clover leaf structure. Now, the clover leaf structure has 3 permanent loops, 3 fixed loops and 1 variable loop. Okay? And it is read from the 5' to 3'. If you look at the 5' end, it has a GGG chain. And if you look at the 3' end, it has a ACC end. Okay? Starting from the 3' end, this end is the amino acid binding site. Amino acid will bind here. Based on what? Based on this structure, the anticodon loop. So what I'm trying to say is, if the anticodon loop has UAC for AUG, right? This is the anticodon. If it has UAC here, then methionine is going to bind to this end, right? So if it is UGU, valine will bind to it, okay? If it is GUG, cysteine will bind to it. So depending on the anticodon, the amino acid is going to bind. So we have a total of 64 codons, out of which 61 are sense codons. These 61, each codon can produce an anticodon. Based on that anticodon, the amino acid will be attached. Okay, now, then the anticodon loop is known as the code recognition site or loop 2. There are 2 more loops, the D loop and the T loop, correct? The D loop contains dihydrouridine, which is an unusual base, and here amino acid transference enzyme will attach itself, which will help in the formation of the charged tRNA, okay? And the T loop has a pseudo-uridine base, which will help in the attachment to the ribosome, okay? And now, this whole structure will be able to put in amino acids based on the code, right? It will be able to read the code, put in the amino acid respective to the code. When we do transcription and translation in detail, it will become more clear to you. But I hope the structure of transfer RNA is okay. Moving on to the last one, which is messenger RNA. Messenger RNA is formed when DNA is transcribed. When DNA is transcribed, its 3 to 5' strand is read by RNA polymerase and an mRNA is produced. This mRNA is your is going to go into translation, correct? The mRNA, obviously the initial mRNA that is produced, the 5' to 3', contains intronic segments. Okay? So, what is done is, hold on. The introns will be removed and the exons will be joined in the next step. This step is known as splicing, which happens only in eukaryotic genome, correct? Once spliced in eukaryotes, this particular mRNA will be modified or processed. How? One, a methylated cap containing a guanine is added to the 5' end. This helps in ribosomal attachment. Okay. And at the 3' end, a poly A tail is added. Other than that, if you look at the internal structure, almost all mRNA start with AUG, which is your initiator codon. So this is the codon which initiates translation. So AUG, which codes for methionine. That means all proteins will also start with the first amino acid being methionine. Okay? And then it will always end with any one of these three, UAA, UAG or UGA. They are your terminator codons which help in stopping translation. Okay? Everything starting from AUG up to the last codon, before the last codon, is the genetic sequence which will get translated. Correct? Which will get read and a polypeptide will be made from it. Now, if this segment, the gene sequence segment, codes for only one polypeptide, then it is said to be a monocistronic mRNA. Okay? So one polypeptide from one mRNA. If you get more than one polypeptide, then it is polycistronic. For example, the one that codes for histone, it has 30 odd enzymes which is coded from one particular mRNA segment, then that becomes polycistronic. Remember that prokaryotic mRNAs are usually polycistronic in nature, whereas most eukaryotic mRNAs are monocistronic in nature. I hope the type of mRNAs was clear to you. The more we delve into the topic of molecular bases of inheritance, this will come in very, very handy. So please go through the video. Let me know if you have any doubts. Thank you.